Let's turn to the more refractory space, and ESMO was just recently, probably the biggest set of data was around belzutifan. There was some monotherapy data from the phase 3, and then also some combination data. So maybe I'll ask you just sort of each to comment. Tian, talk about LITESPARK-005, belzutifan versus everolimus in a refractory setting, mostly third and fourth line largely, but sort of a typical refractory kidney cancer population. What was your initial take on the data?
Positive trial, right? There was a registration trial for belzutifan monotherapy in the refractory setting. It improved progression-free survival compared with everolimus. And so I do think, do I wish it was compared against a different control? Probably, but I think that has certainly activity in refractory settings. And to me, we have to be somewhat selective about the patient population. I would want to see a patient who is a little bit more indolently progressing and seems more angiogenically driven to select them for belzutifan monotherapy.
So in all the trials, belzutifan, the responses have been a little bit delayed, more delayed than a TKI. I don't know that trial reported median time to response, and from a different trial, I have like a 3.6 months number in my head from a different trial of belzutifan, something in that range, which is probably a little more than TKIs. And when you say angiogenic driven, how do you know that?
Hard to tell, right? I don't know that we know. Maybe you'll tell us how. I mean, the gold standard would be to send transcriptome data, but we don't send that on everybody. But it's sort of the clinical gestalt of slowly progressing. Maybe they've had responses to prior IO-TKIs or TKI monotherapies in the past, and they're now slowly progressing. But I do think of all the data that was at LITESPARK-005, it is a very well-tolerated agent, and patients do have good quality of life on it. So I think it's an easy treatment to give.
And especially for that more indolent patient that hopefully could be on it for a while. Right, and if we're not curing patients, then tolerability is important. So David, what did you think of the LITESPARK data?
So once again, I agree with everything Tian said.
I need to disagree more.
I know she has. Well, she's usually right. It's just, if we had Tony here, maybe it would be different. But so building on what she said, I think it's proof of principle that HIF2 is an important target, and that was debated 10 years ago whether that was a target. But when you think about the number of patients who carry a VHL mutation and the response rates with belzutifan, there's a gap between those two, which I think is somewhat exciting in the sense, Tian mentioned one of those opportunities, which is with selecting patients better, you might be able to improve outcomes. That's what the early studies showed. I mean, a PFS of like 14 months in the trials that we did in phase 1, those are better selected patients by definition. We need to find out how to do that. I think we can based on tumor-directed markers, because this is a first exciting tumor-directed therapy. Right. We need to come up with the EGF marker or the BRAF marker for this, and there is potential. There's at least potential there. We have to insist on that research because half the patients didn't get much benefit at all from the drug. The other thing I think we could do is potentially, once we have a marker, is bring the drug up earlier, because as Tian said, it's a pretty non-toxic drug. Patients like being on it when they switch from a TKI to, it's like, right, well, yes, hypoxic water, but yes, so that's exciting, but also trying to fill that gap between the 80% of patients who carry a mutation and 20% response rate. What can we add to belzutifan or other HIF inhibitors to increase the response rate? And those targets may exist, and we should pursue them.
So before, and Hans, I'll get to you in 1 second. So you mentioned combination. There was also data at ESMO, belzu-cabo, in a refractory setting, 31% response rate, and in a frontline setting, a 70% response rate. Right. Select patients, small numbers, all the caveats. You were part of at least one of those cohorts. So what did you think? Do you think those are they surprising, exciting? I mean, 30% in a refractory setting, isn't that great, right? Belzu alone's 23%. Right. Cabo alone's probably in the 20%, so it doesn't even appear additive. What do you think?
Well, I think if you do it if you do that combo in a VEGF naive population, you're going to see exciting response results, but you need to do the randomized trial to figure out what's
We've been burned before by single-arm response rates that are nice and shiny, right?
Yes,
we've all been down that road.
Right, so do the trial is what I would say. And is it worth doing? Absolutely, and if that trial was positive, one of the LITESPARK trials, I forget the number, it would be an incremental improvement. But I guess for patients, which is good, but what I'm talking about is bringing HIF inhibition up front as a single agent, not as a combo. You have a drug that's active, relatively clean. I would prefer not to dirty it up with something that causes lots of side effects, because then it loses, to Tian's point, it loses one of its better components, which is it's clean.
So you think there could be a role for belzutifan or other HIF inhibition monotherapy in select frontline kidney cancer patients?
Yeah,
Not to put words in your mouth, but
not frontline, VEGF naive.
okay.
Yeah.
So in your ipi-nivo failures,
Correct, or PD-1 adjuvant failures.
okay.
Yes, in the right genetic background to
Yep.
building on
Yep.
Tian's point.
Okay. Hans, you're shaking your head.
VHL
no.
mutated,
Oh.
PBRM
I
mutated.
thought we were going to get disagreement finally on this panel.
Well, which is,
So Hans, what do you think? Both monotherapy, the LITESPARK data as it stands,
Sure.
and then sort of the future of.
Sure. So I'm glad it's here, quite frankly. When it works for patients, it's unique. It's a real game changer, especially for patients who are washed out from TKIs. The quality of life for a targeted therapy is unmatched. And yes, some patients with some structural lung disease, for example, may have hypoxia risk, anemia, again, all of these are very easy to manage. Coming to, taking a step back, I'm sometimes a little bit of a biomarker skeptic, so to say, but finding a biomarker for belzutifan will have one of the biggest impacts, I think, for patients and the quality of life. I think it's critical to do that because the drug either works or it doesn't work. So PD rate is very high, right? So there's very little overlap. Who are the patients? I think patients who are probably very close to the VHL pathway. For example, I had a VHL patient, numerous kidney lesions. She was on belzutifan, there was one breakout lesion. And so we had that resected. The lesions around it that were shrinking on belzutifan, they were all beautifully differentiated low-grade tumors, and the one that actually continued growing was a high-grade, fully differentiated, right? So I think it's going to be somewhere in that realm of the better differentiated, closer to the VHL biology-leaning tumors, and the question is how do we identify those? And they were, by the way, HIF2 positive tumors, right? So it wasn't like they weren't having HIF2 in the nucleus. So it's a tantalizing compound, I think. It's not curative, but palliative. Coming back to David's point, I have a patient who had the chance to go on a belzutifan trial right after failure of nivo-ipi. He's now 3 years on it. In fact, he had a CR, and he's happy collecting honey from his bees without any side effects. So it's
On the
doing really
commercials,
well.
I mean.
yes, right.
Go ahead.
No, I was just going to say, building on that, two things. So one, the HIF2 story is real. So Bill Kalin doesn't need to give back his Nobel Prize, which is great. But people didn't think this target could be drugged.
Yeah.
And that's a really exciting story for drug development, not just in kidney cancer, but in other tumors. I mean, targeting it, I don't think people realize, and Lisa Pickering, who gave an excellent discussion, like driving this home. This is not a TKI. This is targeting a transcription factor. People said we couldn't do it. The UT Southwestern team figured out how to drug the pocket, and you have activity.
Do you think belzutifan can replace TKIs? Will we be sitting here in 10 years if we're not retired? And we're going to say, well, how silly we gave people TKIs. Those were things were toxic and not really well targeted. Hans?
I actually don't think so. So coming back to what works, what doesn't work, and maybe like leaning towards that these are VEGF-producing tumors, I actually don't think we're going to find that. So, for example, I don't see typically, dramatic decreases in enhancement on a scan. So it's not really like devascularizing tumors clinically. And I had some patients who did well on TKI before, they got the opportunity to go on belzutifan, it did nothing, and they went back on a TKI and beautiful responding. So I don't think it's going to be
different.
They're different.
Yeah.
They're different period, and I think we really need to invest in how to identify those patients. And I think that would be very impactful work that needs to be funded either by industry or government.
Right, and the last thing I would say is the endpoints are cool too. I mean, the endpoints on that trial are long-term endpoints. We're going to have discussions about duration of response, landmark PFS, hopefully long-term survival. And that's also good for our field, is our patients living longer. We have to keep measuring their outcomes, not just efficacy, but quality of life and toxicity, because the improvements in quality of life were one of the best parts of that LITESPARK-005 study, I think.
What a great story, right, from bench to bedside, as you've highlighted. I mean, our scientists at UT Southwestern from 10 years ago, now to actually affecting patient care and getting into our patients with kidney cancer. I mean, it's a tremendous just
The
bench
story
to
we
bedside.
talk about all the time,
That's
but
right.
so rarely happens.
That's
Right,
right.
and,
Yeah,
and
really.
because so many people
Right.
said it couldn't be done, pharma didn't want to invest until Peloton, which is also a Dallas operation, showed they could do it.
Yeah.
And hopefully that'll have impacts for other
Well,
transcription
and there are
factors.
other companies, obviously, that make HIF inhibitors, and they may
Yeah.
have different pharmacodynamics and different this and different that. So it's really, it's like sort of the first TKI. It's sort of the sorafenib of TKIs.
First in class.
I don't think
Maybe
they
they
want to
don't.
go with
They
that.
don't want to
camp.
be the sorafenib.
Maybe last thoughts on belzutifan. There wasn't a survival signal, right? So the medians were different, but there was no survival. And it was a pretty mature, I think there was 50, 60% of events. Does that matter? Does it just not matter because PFS and well tolerated? Does it not matter because we all think it's going to be used earlier anyway? And so this is, do you care about the survival in 005?
I think it's a step, right? So, I mean, obviously we care if agents are able to help our patients live longer, but having something to delay progression, and to your point earlier, if it's a palliative therapy later lines, and well tolerated, then that's a win.
Okay. David, were you?
I mean, I think it's the reason we have to insist on selection.
Yeah.
As a group, as a community, we have to say, you got to do better than this. So we get OS. I think it might impact, not just approvals, but potentially reimbursement, particularly outside of the US. And if you can't get OS with belzutifan in the later line, what does that say for your second line, your first line, and your adjuvant approaches?
You think it'd be more likely in a refractory setting and less likely as you move earlier?
Well, you could argue with that both ways, but I think OS is harder to get earlier because there's so much other salvage that can be applied at that point. So for example, if you look at the 012 study where belzutifan is being looked at, I think in the triplet combination, it could add to PFS and not OS, let's say, and then what? That might have significant impact on how those regimens, triplets get used.
Agreed.
Yeah, I think LITESPARK-005, it would have been difficult to be honest with you, to show an OS benefit for a patient population that is so selectively benefiting from it. There isn't much overbleed. You don't move the majority of the population. It's not like immunotherapy that gives a little bit benefit to everyone. It's
Everyone?
well, I mean, the medium was moved on checkmate 005, right? The medium was
Yes.
moved on checkmate 005 with a
Yes.
response rate of 20-something percent.
As always, you're right, Doctor.
So the response rate was 20-something percent. There isn't much overbleed. So it's going to be this little. So long term, you may see a little differentiation of the curves. You won't see it now. And knowing that the responses can be quite durable, I wouldn't be surprised eventually you will see something on these curves. It may not be the measure that we normally apply to OS curves, but I do think there's undoubtedly a patient population that will live longer because they're doing well on belzutifan.
Yeah, sure.